Okay, hello everyone. So today I will be talking about some of our recent work on error correcting codes for DNA storage. This is joint work with my PhD student Rami Khabaz and my colleague Mark Antonini. So as probably you already know by now, the DNA storage pipeline is prone to errors in various stages of this pipeline. And these errors occur in the form of deletions, insertions, and substitutions of nucleotides. And as it was mentioned in the previous talks, error correcting codes are one way to mitigate these errors in DNA storage. So I would like to stress that what error correcting codes can offer in DNA storage are the following. First, improved reliability, because you can increase the probability of retrieving your data successfully with error correcting codes. Second, you can reduce the coverage requirements because you would need a smaller number of reads to recover your data with error correcting codes. And third, you can correct what I call systematic errors. I don't understand fully the biochemical aspects of this, but I've seen data from colleagues where you have systematic errors, meaning the same error pattern appearing across all reads for a given oligo. And in that case, no matter how much you read and accumulate reads, you will not be able to correct these error patterns. But if you do have an error correction code, then you can address this over an individual sequence. And last but not least, this was briefly mentioned also, it can contribute indirectly to reducing the costs of DNA storage. Why? Because today, one of the reasons why synthesis is very expensive is because companies are putting so much effort to produce almost perfect oligos. But what if we work with less expensive and more noisy technologies and compensate for this excess in noise using error correcting codes? Then these cheaper technologies can become viable. And a common design strategy is using an inner code and an outer code to use error correction in DNA storage. So with the outer code, you basically generate redundant oligos in your system. And with the inner code, you add redundancy within each oligo in your system. Now, ideally, the inner code should be a short code capable of correcting deletions, insertions, and substitutions of nucleotides at the sequence level. Why short? Is because the DNA oligos are short due to synthesis limitations, whereas the outer code could be some longer code designed to correct lost oligos or some residual errors from the inner code. What is the state of the art when it comes to this? So there are many existing codes which are suitable for outer codes, such as Reed-Solomon codes, LDPC codes, Raptor codes, etc. But there are very few practical codes that are suitable for inner codes, and this is an active area of research. Why is it an active area of research? It's because you cannot use any off-the-shelf code to correct deletions and insertions, and this was mentioned in the previous talk. So you cannot use an LDPC code to correct deletions and insertions, unless you have many reads and you do something, but in the strict error correction sense, you cannot do it using LDPC, Reed-Solomon, or Raptor. Okay, so this is also a very fundamental area in information theory. So there has been a lot of works in the recent years on designing codes for correcting edit errors in information theory. This great theoretical work has not translated into practice yet due to some limitations, such as being impractical for short sequences. For that reason, the works that have actually implemented this inner and outer code decoding strategy have not used any of the codes I mentioned above. Instead, they heavily rely on the inherent redundancy of sequencing. So through repeated reads and consensus algorithms, they address deletion and insertions, but even in some works, whenever they end up with reads which are not of the correct length, let's say one less or one more to deletion and insertion, they simply discard them. And we think this is wasteful. And there's much that can be done using error correction to address these cases. There are few exceptions to this. So there are actually some codes out there which are practical for DNA storage and work for short sequences, such as HEDGES is one of them you may have heard of. Our contribution is in this line of work. So we propose a novel code that can correct deletions and insertions and substitutions in short sequences. And we call this code marker, guess and check plus codes, MGC plus for short. So I will try to explain how MGC plus codes work, the encoding and the decoding, very briefly. I will not go into the technicalities. So this is going to be a simplistic explanation. So starting with some digital files, we first fragment this into shorter fragments, and then we use any outer code to generate redundant fragments. Our contribution is in the inner code part. So now, given a certain oligo, or at this point it's not an oligo yet, it's a binary sequence, we add some redundancy to this binary sequence using Reed-Solomon codes. But I just told you that Reed-Solomon codes don't work for deletions and insertions. That's why there is a part in the decoding, which I will later explain, which will make Reed-Solomon codes viable for correcting such errors. And this redundancy is divided into what we call guess parities and what we call check parities. And these check parities are going to be further encoded using an additional layer of error correction. And then we're going to map this to DNA using DNA coding. It could be as simple as 2 bits, like one DNA symbol for every 2 bits. And then we're going to introduce some markers in this sequence, periodic markers. And in the end, we get our coded oligos, which looks something like this. Now, the interesting part is the decoding. So now let's say we have one oligo and it's affected by errors. The first step is to decode the check parities, and I will explain shortly why some parities are for guessing and why some parities are for checking. So we first decode the check parities, and then we use a trellis-based estimation algorithm to infer if there is any offset in each of these marked blocks. So there are markers. At the end of each block, we have a marker, so that makes a marked block. And we use a trellis-based estimation algorithm to detect if some of these segments did have an offset. When I say offset, it means they had a deletion or an insertion. And then we output this offset pattern. Here, -1 means that there was the number of net deletions was -1. Now, for the blocks which have a non-zero offset, we're going to map them to an hypothetical erasure. And for the blocks which we estimate that no offset occurred, we're going to use their actual value. So now we have a mapped sequence which has some erasures and some substitutions also. So this is how we map or reduce the problem of correcting deletions and insertions to correcting substitutions and erasures, which are easier to deal with. And Reed-Solomon codes can deal with erasures and substitutions. So now we use the check parities to see if our guess here on the offset pattern is true or not. If it is true, we're done. If not, we generate more informed guesses based on the estimated pattern. And then for each of these patterns, we also check if they are valid or not. If there's any match, we're done. If not, we just mark the sequence as an erasure, so as a strand loss. So in the end, the output of the inner decoding is going to be a bunch of sequences. Some of them are marked as erasures, and others are not, but they could still have errors. So that's what the outer code deals with and then corrects these. So this is the high-level idea of the code works. So what can this code achieve? So first, I'm going to show you results for the inner code performance alone as a standalone. So there's no outer code here, and there's no multiple reads per sequence, just the standard coding theoretic performance setting. So here we have 1 million random messages of length 176 bits, and their error rates are applied based on research experimental data. And we're comparing here with hedges. We're showing the probability of decoding error versus the edit probability and the average decoding time. And here the observation is that we can achieve a better overall decoding performance in terms of probability of decoding error, also in terms of code rate, because we are able to achieve better code rates, and also, very importantly, in terms of decoding time. Because with the MGC plus codes, we are able to achieve decoding in the order of milliseconds per frame, and this is on a personal computer of my student, so it's not any fancy hardware or anything. So this shows the comparison of the inner code, MGC plus versus other constructions. So now let's go into a second setting where we have now an outer code, so we're actually encoding a file here. So we have an outer code, but we're assuming we only have one read per oligo. So this is the least. So only one read per oligo, but the error rate is only 1% also. Let's see what we can do here. So we can, of course, configure the inner code in many ways. And we're trying to find out for each configuration of the inner code, how much redundancy we need from the outer code to be able to recover this file successfully 100% of the times. When I say 100%, it means 100 out of 100 files, because we're generating random files. So here, for each inner code, we have some outer code rate which can achieve this 100% success probability. And then we are looking for the optimal trade-off now, like what is the optimal balance between the rate of the inner code and the rate of the outer code that maximizes the information density. And here, a certain configuration gives us 1.13 bits per nucleotide. And this is, again, with only one read per sequence. The next question is, what if we have multiple reads per sequence? So here, in this setting, we have multiple reads per sequence, and we have a 10% error rate now, not a 1% error rate. So the simulation setup is kind of the same. Now the inner code is an MGC plus code that I explained, and the outer code is a standard Reed-Solomon code. We're generating multiple reads per sequence, and the number of reads per sequence follows a Poisson distribution with some parameter C. So it's not exactly C reads per sequence, but the average is C, and C is the parameter we're going to vary. It's like the coverage depth, what's called coverage depth in certain papers. Okay, so here I'm showing you results for each value of the coverage depth. What is the optimal balance between the inner code and the outer code that allows us to achieve 100% success rate in terms of decoding? Intuitively, as you increase coverage, your coverage depth, you are able to achieve better information rates. That's very intuitive. But it's also important that for this particular setting, for coverage depths for less than 9, we were not able to achieve 100% decoding without using an inner code. So this shows why the MGC plus code can be important as an inner code in DNA storage. To sum up, I talked about the MGC plus codes, which are promising candidates for inner codes in DNA storage. They are efficient for short block lengths, and they improve the reliability of the system, and they can reduce the number of reads needed for decoding, which is also interesting. Now, all the results I showed were based on simulations, numerical simulations, using mathematical models. The next step is to consider more realistic simulators, and some simulators were discussed today. Obviously, there are many simulators out there, but the one we're trying to consider is the one called Digital Twin for DNA, which is developed by Robert Grass's group. So the next step is to test this on this simulator before going into the following step, which is obviously doing wet lab experiments. But since wet lab experiments are expensive, as you already know, we want to try to optimize things as much as possible numerically before taking that step. And before I conclude, I just want to mention that we're organizing an autumn school on DNA data storage between 10 and 14 November in the coastal town of Cargèse in Corsica, France. This school is intended for PhD students and junior researchers who are interested in DNA storage in general. And to participate, you will have to submit an abstract. The QR code of the website is here if you want to check. And if your abstract gets accepted, the PLPR Molecular Archive project will cover all costs except the travel costs to Cargèse. So the accommodation, the meals, the stay will all be covered for accepted abstracts, but you will need to cover the travel costs there. So that's all, and thank you.
Two? Okay. So yeah, one question, and then the rest questions can be asked during the coffee break.
Thank you. You talk about at the end the experiments being expensive, and if there was an experiment you could do and to get some info, what would it be? The first one you would like, or the info you would be happy to have?
The info that I would like to have is the actual error profiles that happen in practice. So there are some works out there on this, and this is the basis for many simulators. But I would like to go deep in that. Like, it's not about what % error rate to expect. It's also the statistics of these errors. Like, are they uniformly distributed, or are they localized in certain sequences? Because you can say the error rate is 5%, but that could mean 1,000 things in statistics. It could be 5% in only one sequence, or I don't know. Like, the statistics is really important. So that's the information that's really useful for error correcting code.
Thank you.
Thank you, Serge. Thank you. Round of applause.